Hi, my name is Michelle Mielke, and am a professor of epidemiology and neurology at the Mayo Clinic in Rochester, Minnesota. In terms of my training background, I received a Bachelor of Science in Neuroscience from the University of Pittsburgh, and then I went to get my PhD at the Johns Hopkins School of Public Health, Department of Mental Health, where my PhD was specifically in psychiatric epidemiology, and I was a pre-doctoral fellow on a training grant. My thesis focus was on vascular risk factors for Alzheimer's disease. I then stayed at Hopkins, where I had two one-year postdoctoral fellowships, one in the epidemiology of aging, and then the Lydia Gillespie Fellowship in psychiatry. After that, I stayed on faculty first as an instructor and then assistant professor at Johns Hopkins School of Medicine for about 5 years, and then moved to Mayo Clinic, where I've been for almost the last 10 years now. So, a question: how did I become interested in Alzheimer's disease? Well, I had always been interested in the brain and really how such a concrete structure results in such abstract thoughts and feelings. And however, when I went to the University of Pittsburgh as an undergrad, my goal was really to become a behavioral neurologist. I was going to major in neuroscience and psychology, and I really had no interest in research. However, I did have work study, and so I thought it would be a good idea to get involved in some research, which would look good on my CV for medical school. And so I ended up working in a lab that was examining nicotine self administration in rats. I was extremely fortunate in that the PI of the lab, Dr. Anthony Kajula, really allowed me to get involved with research. And by my third year, I was planning and running some of the studies for his grants. And I found that I really love the research. Originally when I thought about research, it was like, oh, how am I going to come up with questions? I don't think I can do this. But I found that it was really invigorating, and I love to look at these questions and continue to learn. Um, I had a snafu with a loan my fourth year, and I had to resubmit it. But in the meantime, had to get another job. And so I ended up working as a nursing assistant in a nursing home. And it was there that I really began to fully realize the impact of dementia. And I had all these residents that I helped throughout my shift, but I really didn't have the time to spend with those dementia patients that I really wanted to. And then around that same time that I was working in the nursing home, I also had a class on the neurobiology of learning and memory. And I wrote a paper on the effects of hormones on the brain. So I started to think about sex differences. And this combination of experiencing and working with the patients and the residents, as well as my growing interest in understanding Alzheimer's disease and sex differences, led me to research in Alzheimer's disease as well as in epidemiology. It took me, I was considering going and getting my PhD for neuroscience or epidemiology, and really I decided I wanted to work with people as opposed to animals. I have a lot of research interests. Maybe I don't say no enough. Um, but this it general boils down into true primary foci for my research. One is fluid-based biomarkers for Alzheimer's disease and related disorders. And secondly, understanding sex and gender differences in the development and progression of Alzheimer's disease and related disorders, as well as other aging-related conditions. For fluid-based biomarkers, I focused on AD-specific biomarkers such as plasma phosphatide 181, markers of neurodegeneration such as neurofilament light chain, as well as inflammatory markers and sphingolipids. In 2018, we published the first paper examining plasma p-tau 181, showing that it was associated with amyloid and tau PET. Now, a next step and what we are currently focused on is to try and understand how these biomarkers look in the general population. So, subsequent papers that have come out on plasma p-tau, for example, as well as amyloid and NFL, have primarily focused on clinic populations and more specialized populations. And so, as an epidemiologist, I'm really interested to see how these can work in the community, particularly since dementia patients are going to be diagnosed and seen by primary care physicians. Do these biomarkers work as well as in these specialty clinics? And what might be some factors that are associated with these markers that could affect the interpretation? Because in primary care, there's going to be a lot more comorbidities and other aspects to think about. Um, Second theme of my research is focused on sex and gender differences and sex specific risk factors. Although the incidence of Alzheimer's disease does not differ by sex in the United States, the pathway of how men and women develop the disease and the risk factors can differ. So, we are working on understanding sex differences and risk factors, looking, for example, at vascular risk factors. And then, in addition, trying to understand the effects of hypertensive pregnancy disorders and gynecological surgeries on risk of dementia in women. Historically, sex specific risk factors have not been looked at as closely. Right now, we have an ongoing study of about 3,000 women in which we are looking back in their medical records to record all information about their pregnancy, any gynecological surgery, whether it's a hysterectomy or removal of the ovaries, information about infertility, age of menarche and menopause, and then also recording any type of hormone therapy that has been used. So, whether it's for contraceptive reasons, menopausal hormone therapy, or even for breast cancer treatment and prevention. Um, and ultimately, our goal is to develop a lifespan model of these risk factors that can better predict the risk of dementia in women. And then after that, we hope to do the same for men, such that if we develop sex specific models, that will be more predictive um, for both men and women than if we just combine women and men together.